So yeah, welcome back. Yesterday we completed lipids, and now we're gonna go towards protein, and after that nucleic acid and water will be covered combined. Now let's just start. So we're live, yeah. Now remember from the first class that I told you that protein is not a polymer. Is not a polymer. It is rather macromolecule. Because it is not necessarily made from you can say similar subunits. The subunits are different, which combines to form a protein. Now the monomer here is now the monomer of protein is called amino acids. Yeah, amino acid is not a single thing, it is group of amino acids. In this case there are 20 amino acids. Yeah, there are essentials and non-essentials, 1, 11 and 9 respectively. So the essentials ones are the ones that we need to take outside, from the outside of body, whereas the non-essential ones are produced inside. Yeah, so before I should start with explaining the structure and stuff, or maybe I should first explain the structure of a amino acid. So the basic structure of amino acid is this. C-double-bond-O-O-H. Now this is a typical amino acid. This whole structure is the main skeleton, and this same structure is, or this structure is same in every amino acid, this structure, but the only difference in amino acids is this variable R group. The differentiation in amino acid is because the variable group, which can be CH3 or H or etc., different types of groups. Yeah, but in this case I'm just gonna discuss two, for example. You need to memorize this structure of amino acid, the main skeleton I'm talking about. For the A-levels, you don't require to memorize the whole structures of amino acids. You don't need to remember the all 21s, but I should just name for a reference here. So alanine, phenylalanine, isoleucine, proline, leucine, tryptophan, methionine, valine, asparagine, serine, cysteine, glutamine, glycine, tyrosine, threonine, serine, and aspartic acid, etc. There are like 20, yeah. And the main difference is this variable group. For example, we just take an example of glycine. So glycine is an amino acid with this structure. If we talk about another structure, which is alanine, so glycine is the smallest amino acid because it only contains H on both sides. The variable group contains H, only hydrogen, whereas alanine, for example, the structure of alanine is and let me just give you another example. For example, serine, which is another type of amino acid. So you can memorize these three basic ones just for your easiness. So the thing is, you can see here that the main skeleton, the one which is this is same in all of them. This is same in all of them, you can see here. But the only difference here is this variable group, and this variable group, which leads to different properties of these amino acids. Now, amino acids bonds covalently with each other via peptide bond to form polypeptide. Polypeptide, yeah. If two amino acids join together, this is one amino acid and another amino acid, this is called dipeptide. If three, for example, 10 peptides are joined, or more than greater than 10, if the amino acids are greater than 10, then it is called peptones. Let me just show you how they bond here, yeah? So NH, CH, and for example H here, and C double bond O and OH. So this is a typical amino acid. We take another similar amino acid and place it here. Now what's gonna happen is that hydroxyl group reacts with hydrogen and water is eliminated, and the bond is formed, which is called peptide bond. So this is how a peptide bond looks like, where carbon is directly connected to nitrogen. So this is a dipeptide. Both alanine, both have glycine. Gly. This is the way of writing a sequence. So we have discussed the bonding here. Now let me just tell you one thing which was very interesting to me when I first read it. So for example, there are 26 letters in English language. D and E, blah blah blah. Now, with the combination of different letters form different words. For example, hello, hi, sup. These are different types of words that we use in English language, or this might be a normal day chatting, or you can say, but the thing is, these letters, 26 out of those 26 letters, we use different combinations. Millions of combinations are possible because of those letters, and the combination of those letters form different kinds of words, and the possibilities are in millions and billions. You can expect any possible combination from letters. Now, but the thing is, words, or you can say the words in English language, can only extend up to maybe, okay, let me just search about it. So yeah, the longest word in English language is a medical term with 45 letters arranged in different arrangements, whereas there are thousands of amino acids which are in quantity of 20. There are thousands of amino acids, thousands and thousands of amino acids that can be sorted in a sequence to form protein. So the longest letter is the longest word is 45, but the longest protein can be of thousands of amino acids being bound together. So there are, I don't think so, I don't know how many possibilities are there, but there can be billions of possibilities of amino acids being in a specific sequence to form a specific protein. So for example, we take with the three that we have learned. So for example, glycine, serine, and then alanine. So these three combines to form a single protein. For example, protein are usually longer, but for example, and another combination could be glycine and serine. Another combination could be serine, and glycine, and so on. So there are numerous possibilities here. Anyways, so this is what amino acids. One more thing I should tell about amino acid is that you see this main skeleton here. The thing to notice here is that there is a acidic group and an alkaline group, making it a kind of buffer compound. Or if you don't understand what buffer means, I will explain it in another chapter, but let's just give you a brief introduction about that. So buffer is a solution which resists the change in its pH. So what happens in a buffer solution is, for example, this is a solution of amino acid. Now here we add a good amount, or you can say substantial amount of acid. And acid contains, or you can say HCl, which is acid basically. So acid contains high concentration of H positive ions. So if we add hydrogen ions or acid in the solution, due to the presence of H positive ions, they will move towards the nitrogen, which is alkaline, and this nitrogen can accept hydrogen atoms. So these hydrogen atoms will travel here and bond with this nitrogen, giving it a positive charge. So this is what happens when you add an acid. So this lowers, so this maintains the pH of the solution and prevents it from lowering down. Now if we add an alkali instead of an acid, which contains hydroxide ions here, or you can say any hydrogen accepting you can say compound, when the concentration of that increases in the solution, increasing the pH, what happens is the hydrogen atom from the carboxylic group reacts with the hydroxide group or hydroxyl group, forming H2O, which is neutral. So this again resists the increase in pH and maintains the pH. So this is the buffer quality of protein. So protein has a buffer quality. So the thing is, I should just you can say review what we have studied till now. Protein are polymers of amino acids, and amino acids are basic units from which protein are made up of. 20 are more commonly found amino acids in proteins. Plants can synthesize all required amino acids, unlike animals. So animal can make some, but they have to take others that are required in their diet, which are called essential amino acids. So we have talked about the structure, we've talked about the main skeleton, we've talked about the types, different types, the three types that I've told you, the different names that I've told you. Amino acid is composed of alkaline group, amine group, and acidic, which is acidic group, and the carboxyl group, or you can say the amine group, which is the alkaline group. Both of these groups are attached to central atom in the main skeleton of molecule. A variable R group is bonded to the central atom, carbon atom. It varies in every amino acids and defines chemical properties of amino acids. R group makes up the side chain, which is the reason of amino acid being different from each other. The larger the chain, the variable one, because the other ones are similar in the amino acids. So the larger the side chain, the more you can say denser the amino acid. So I've told you what exactly happens as a buffer solution, but in the deep concept in chemistry, there is a concept of Zwitter ion. So without explaining, I should just tell you what a Zwitter ion looks like. So this is a Zwitter ion of amino acid. So just on the surface, I should just tell you that the Zwitter ion is just a stable form of the protein in aqueous solution. So if you pour protein in aqueous solution or dissolve it, this is a ionic condition that protein will get into, and when you add an acid, the H positive ions will react, when you add an acid, the acid will be added to the oxygen, the negative side of the Zwitter ion, and if you add the hydroxyl group, the H positive, the extra H positive that is on the nitrogen side will react with the OH to form water. So this is Zwitter ion concept. Now, we have also talked about the condensation of amino acids forming peptide bonds. Different type of peptide bonds form between the amino acid. Here we have talked about it. And also I should just tell you that one thing I should tell you that it is a question that comes mostly in MCQs, that you should remember that this is very important. So the number of water molecules that are released on the condensation process is n minus 1. This is the formula of water molecules. So n is the variable number. For example, if 20 different amino acids get into the condensation reaction, the variable is going to be, the n is going to be 20, and the water molecules that are released are going to be 19, and the bonds that form are gonna be 19. This is the number of bonds and the water molecules that are released. Now this is a very basic thing, and usually I shouldn't be telling you this, but a lot of times when you are solving MCQs in a hurry, you just do that question wrong. Now, hold on, let me just drink water. Now, we should talk about the structures, or you can say the structures that protein goes into. So, remember I told you that polypeptides can be thousands of amino acids being been in condensation reaction to form a protein. So there are 4 main types of, or you can say 4 main structures of protein. Number 1 is primary. Number 2 is secondary. Number 3 is tertiary. And number 4 is quaternary. Now, what primary is? So primary structure is nothing but simply the sequence of amino acids. So if you ever see, for example, alanine, and and so if you ever see polypeptides, or you can say in a polypeptide you can see the number and the type of amino acid being bonded together via condensation reaction, the structure is called primary structure. There is no bonding involved in this structure, remember that, except for the covalent bonds. Now, what secondary structure is? So, secondary structure involves hydrogen bonding, yeah? Nothing more than hydrogen bonding. You should remember that, because I had a lot of difficulties in understanding that, but in summary I could understand it like there is no more bond except for the hydrogen bonds here. No kind of sulfide bridges, no kind of interaction between R group, no hydrophobic bonding, nothing. Just simply hydrogen bonds in a single chain. So this accounts for the local interactions in the chain. Now, what happens in tertiary? Now, here is where R groups come into account. So, in this tertiary group, there can be hydrogen bonding among in the chain. Okay, so remember this too. In tertiary, the bonding again is inside the chain. So it could be hydrogen bonds, it could be sulfide dibridge, or you can say sulfide bridge. And this is mainly because of the interaction between the different R groups. So I told you about the CH3 and CH3OH kind of R groups, but there are numerous which contain sulfur and stuff. So there can be different types of bonds between the R groups, but the bonds that form are strictly within the chain. Now, I should tell you one thing as the order of the strength of bonding. So, in the R group, remember that. So the strongest is the peptide bond in protein, then disulfide bridge, and we have talked about this before. Then the stronger bonds are ionic, which can also form between the R groups. Then hydrogen bonds, Then there are local hydrophobic interactions, or you can say hydrophobic interactions. So let me just explain what hydrophobic interaction means for a bit. So for example, A is a amino acid that is non-polar. So that means it is insoluble, or hydrophobic means it is water repellent. So it is bonded to different kind of hydrophilic atoms here, then A, then hydrophilic, then A. So remember what I told you before yesterday, in the chapter of lipids, the property of the hydrophobic compounds. So if this compound is poured into water, what is going to happen is that due to the property of it being repellent to water, it will bend towards, or bend inside the structure, and because this one has hydrophilic properties, this will move towards water, and this will create bends into the structure, where inside there would be hydrophobic, and outside would be hydrophilic. So these kind of interactions, or the bending, the change in structure, which is caused due to the interactions between hydrophobic compounds and water, the different type of structure that forms due to this property is the reason, or you can say the property, which is called hydrophobic interactions. Now, what quaternary structure is, for example, different chains interact with each other or bond with each other, either via a disulfide bridge, hydrogen bonding, or anything else, a bond, or you can say hydrophobic interactions, or with a metal. The bonding of different kind of protein molecules leads to the formation of quaternary structure. So this is what quaternary structure looks like. For example, the structure of hemoglobin, that is a quaternary structure. Now, let me just tell you the classification of protein via structure. So there are three types of protein structures. Or you can say it according to the shapes. One is globular, another one is fibrous, and another one is intermediate. So globular structure is usually tertiary structure. Alhamdulillah. Fibrous structure is usually secondary, and the intermediate is the mixture of the tertiary structures, you can say. Now, I should just explain how a globular structure looks like. So, the properties of globular are that it is compact due to extensive bonding. It is compact, roughly spherical, circular in shape. It is soluble in water, and the non-polar hydrophobic R groups are oriented towards the core center of protein, away from the aqueous surrounding. This is what I explained before. Whereas hydrophilic groups are acts oppositely, maintaining the tertiary structure of and the solubility. So globular structure plays an important role in physiological processes, as they can be easily transported from one organism from one place to another in the organism and be involved in metabolic reactions. So because of its solubility, it contains physiological importance. And if we talk about the types or the things, for example, enzymes, all the enzymes can be globular, or you can, because they are functional. So enzymes can be globular. Hormones, hormones, not really hormones, but yeah, hormones can be protein too, because they are protein in nature. Enzymes, different types of enzymes. The structural protein is basically most of the time fibrous, because it is insoluble. For example, keratin in our you can say nails, the protein in our hair, it is insoluble, so it is secondary structure, which is a you can say fibrous protein. So the difference between globular and fibrous is that, for example, you take the shape of globular protein is roughly circular, whereas fibrous are long strands. Amino acid sequence in globular is irregular, and wide range of R groups, can be anything, whereas in fibrous it is repetitive and a limited range of R groups. Functions, physiological, functional, it is used inside body for physiological functions, whereas fibrous have structural importance. The examples of globular structures or globular, you can say globular proteins, are hemoglobin, enzymes, insulin, immunoglobulin, etc., whereas fibrous includes collagen, keratin, myosin, actin, fibrin, etc. Now, generally globular are soluble in water, and generally fibrous are insoluble in water. Now, there are two things more in this chapter that are explained. One is the structure of hemoglobin, and another is the structure of insulin. But the thing is, I wouldn't discuss it here, because I would discuss these all things in another chapter. The structure of hemoglobin is going to be discussed when we talk about chloride shift in the transport in humans, and the structure of insulin is going to be, we will study it in genetics. Yeah, so we should just leave that here. Anyways, I can tell you about the functional proteins as an example. You can say for digestive enzymes, amylase, lipase, pepsin, and trypsin are the types of digestive enzymes, which are protein. The transport protein involves albumin and hemoglobin. The hormones that include insulin and thyroxine are also proteinaceous. The defense mechanism, immunoglobulin, interferons, are also types of protein. The contractile proteins are actin, myosin, and the storage ones are limbic storage protein or egg white albumin. So I think I've covered everything you need to know in the chapter of protein. The only thing that is left is the structure of hemoglobin, the structure of fibrous protein, which we will discuss in the chapter of cells, in the portion of cell wall, obviously, and the structure of, you can say, the insulin. Now, how much time do we have? Okay, let's just end water part 2. One thing I forgot, sorry. One very important thing here I forgot to mention is that protein are temperature and pH sensitive. This concept we usually discuss in the chapter of enzymes, but I should be telling you in this chapter so that you should remember it in the next chapter or in the upcoming chapters, for example enzymes, that protein are temperature and pH sensitive. Now why does that happen is because protein contains R groups, protein contains different kind of hydrogen bonding, which produces a kind of structure of a protein. For example, this is a protein, and this has a very important function inside the body. This shape of specific protein is due to hydrogen bonds. This is a hydrogen bond, this is a hydrogen bond, this is a hydrogen bond, and this is a hydrogen bond between them. Now, for example, if the temperature increases, these hydrogen bonds will get weaker and starts breaking, leading to the protein to lose its shape. Now, because protein has lost its shape, so does its function. Because proteins, like enzymes, are specific, highly specific to each other based on shapes and complementary to each other. This we will further discuss in the chapter of enzymes, but this concept was necessary for you to be, or required for you to be known in this chapter. Now, we should move towards water. Then all that's left is gonna be nucleic acids. Then we can move towards cells. So water is the most important compound, not let alone inorganic compound. So it's the most important and I should be telling you why is it so important, because of a lot of the properties that water has. So water is the most abundant component in the living cell, and its amount is approximately 70% to 90% depending on different types of cells, but it is a kind of medium of life, because all the reactions that take place in a cell, water provides a medium, or these all occurs in the presence of water. Also, water takes part in the biochemical reactions such as hydrolysis, condensation, also provide raw material such as for photosynthesis. I mean, it is very important as we have studied about the dehydration synthesis and the you can say hydrolysis too, as well as it is important in photosynthesis too, which we'll study in the bioenergetics chapter. Now, why water is so special? Let's just see. So this is a structure of water. The bond angle is 104.5, or you can say 45. It is not necessarily important for you guys to remember it, but if you want, you can. So the properties that water holds are kind of anomalous and different from the pattern that we see elements following in groups. And why is that so? So the thing is that water has electronegative and partial oxygen in water is highly electronegative, leading towards partial negative charge on its oxygen and partial positive charges on its hydrogen and partial negative charges on its oxygen. Yeah, so because of these partial positive charges, there is attraction of it with other water molecules. For example, there is another water molecule here. There's another water molecule here. Another water molecule here. So what happens is these H positives are attracted to partial negative. These partial positive are attracted to partial negatives. These partial negatives are attracted to partial positives. So these are hydrogen bonds that form between water molecules. One water molecule can form 4 hydrogen bonds, one from here, or I should just one from here, one from here, one from here, and one from here. So this special quality of forming so many hydrogen bonds within itself gives water different types of abilities or properties that is very or highly important from the respect of life. Now, what are those properties? So number one property is cohesion, or cohesive force of attraction. Another one is adhesive. Another one is high specific heat. Another one is high heat of vaporization. Another one is, oh, maybe I should just leave it to the last. And another one is ionization. Of water. And the last one is, or not the last one, but anomalous behavior. And the last one is going to be hydrophobic exclusion. So, what is cohesion? Let's discuss that first. So, for example, this is a water molecule. And this is another water molecule. And this is another water molecule. Sorry. So all of these water molecules are connected with each other via hydrogen bonding. So the ones represent hydrogen bonds. So these water molecules are bonded via hydrogen bonds with each other, or you can say attracted towards each other because of hydrogen bonds formed between them. Now what happens is, for example, you take one atom, this atom for example, you take this atom and drops it from the height. So what's gonna happen is not only this single atom is gonna fall, but all the different atoms that are connected with it are gonna fall with it. So this property of not getting or not falling alone, but taking others with yourself, the cohesion or the cohesive force, is what called the cohesion between what am I saying, sorry. So the property of not falling alone but taking others with you is called cohesive force. Now, why is this important? For example, this is a tree and stem and it contains xylem vessel. Now, another property before I should explain you is explaining this xylem, what happens inside xylem, is cohesion and adhesion. So what adhesion is simply hydrogen bonds between water molecules and the molecules of cellulose or wood or whatever you want to say. So for example, these are molecules of xylem or the walls of xylem. So the ability of water to form hydrogen bonds with the molecules of the wall is called adhesion. So the ability to form hydrogen bonds within its same molecules or like molecules or brothers and sisters, for example, is called the cohesion, because they makes hydrogen bonds between themselves, whereas the ability to form hydrogen bonds with other molecules like cellulose and stuff, or you can say the compounds that are present in the walls, lignin for example, that ability is called adhesion. Now why are these so important? So for example, this is a tree and this is xylem in it. Now what happens is, if xylem taking account of the energy used by it to transfer water against the gravity, it uses energy to take via active transport water inside the xylem vessel. Now if this cohesion property does not existed, water would have just gone like droplets. Yeah, it would have been required a lot of energy to take one drop in, one atom in, another atom, then another atom, and then another atom. But what happens is, in reality, what happens is that water or water molecules, if one enters, then the rest enters with it due to the bonds between them. And then if water molecules are at such height and there is a gravitational force which is pulling them down, what happens is they form bonds between the walls of the xylem vessel. So here we have interconnected water molecules going up, and because they are interconnected, or also they have hydrogen bond formation with the walls, this helps them to not get down or to stay here instead of just going with the force of gravity. And because of these two properties, the transportation in plants is very easy, or you can say this helps in the these two properties helps in the transportation of water inside xylem, or transportation of water inside the plants. Now another property is called high specific heat capacity. Now this is really the concept of physics here, but I won't go deep into it. Or maybe what we should do is, as I can see here, the stream has reached for an hour. Why shouldn't we continue this chapter by tomorrow? So we are going to be left with high specific heat capacity, high heat of vaporization, ionization of water, anomalous behavior, and hydrophobic exclusion. So we're going to be left with 5 different properties. Also, this hydrophobic exclusion, I'm going to explain it a bit more than it's required, because you really need to understand what's the chemistry behind this. So these 5 things plus nucleic acids will be discussed tomorrow. So yeah, until then, bye bye.